Hi everyone, I hope I am audible. Hi everyone, I hope I'm perfectly audible and visible. I guess I am. Hi everyone, welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma. I hope you all are doing well and I hope you all are preparing sincerely for your NEET 2026 examination. Hi everyone. So quickly in the chat section, do let me know, am I audible or visible to all of you? Yes. Am I audible or visible to all of you? Yes, please. Am I audible or visible to all of you? Sure? Okay, so welcome to the session, guys. So this is our session, and this is session is basically in the basically we are right now we are continuing the level up series in which we are picking up the most important chapters, and of course the basic one, right? Everything in the lectures will basically in all the lectures we always start from the basic level, from the scratch, and then we move to the right advanced level. So don't worry if you guys are watching this lecture for the first time. Even trust me, you are going to understand each and everything, because today we are going to start the eukaryotic cell. So we are already done with the prokaryotic cell. Today we will start eukaryotic cell. So we are discussing the cell chapter, which is basically the foundation of your biology. I hope you all know that cell is the basic unit of life. Cell is the basic structural and functional unit of life. So of course, if you need to understand the biology, you need to start the biology. You should know what exactly the cell is. So we are done, right? Yes. We are done with the prokaryotic part, and now we have started the eukaryotic part. And trust me, we all are planning different things so that we can provide you the best content, and that we are providing from last 2 years here on this Unacademy NEET English platform. And we are planning to increase the frequency of the sessions as well. We are planning to have the practice sessions as well. So just allow us some time, because this time the planning is going to be detailed, and according to that we all will stick to that, and accordingly we will complete our chapter. So don't worry about that. Okay, don't worry about that. Done? Yes, beta. Girish, you are right. Visible per month, 1 day, like whatever you can say, because we have the batches as well, and trust me, they are going very well. And in that batches we really need to focus a lot, right? We need to focus a lot there. Okay, so that's why we are quite busy, right? We are quite busy because we are giving our level best to them so that they can score really good in NEET 2026. Okay, Chali. So if you guys want to be the part of our batch, so you know that in the description box of this particular video, you guys will get the link for that. Okay, so let's start the eukaryotic cell. Let's start the eukaryotic cell. So students, already we are done with the overview of the cell. Now let's start this part. So when you talk about the eukaryotic cell, you know that U means true. What is the meaning of U here? U means true. And what is the another word that we have here? What is the another word that we have here? It is carry-on. Right? EU is true, and what is the another word here? It is carry-on, and we call it as nucleus. So the cells having a true nucleus in which we are having our genetic material is basically the eukaryotic cell. Is basically the eukaryotic cell. And not just the nucleus, the membrane bound nucleus, but other than that, membrane bound organelles are also there, right? Membrane bound organelles are also there. So this is basically the eukaryotic cell. So if I talk about the evolution, right? So you know that we have prokaryotic cell first, and then we have the eukaryotic cell first. So eukaryotic cell is advanced than prokaryotic cell. Eukaryotic cell is basically having membrane bound organelle. It is having better, I would say, the it is having better division of labor. There are different organelles which will conduct different functions. So this is what we are going to discuss here. Okay, this is what we are going to discuss here. So when we talk about the prokaryotes, we talk about one organelle that is non-membrane bound even, and that is your ribosome. Remember students, in the prokaryotes, we talk about the non-membrane bound organelle, and what What is the name of that organelle? That is ribosome, the protein factory. But in the case of eukaryotic cell, you have membrane-bound organelles, many membrane-bound organelles. Like when it comes to the single membrane-bound organelles, you'll take the example of endoplasmic reticulum, you'll take the example of lysosomes, you will have the examples of Golgi apparatus. Yes, students, right? You will even talk about the microbodies, which are basically single membrane bound. And when it comes to the double membrane bound in eukaryotes, yes, students, when it comes to the double membrane bound organelles in eukaryotes, we discuss about the nucleus, we discuss about the mitochondria, we discuss about the plastids or the chloroplast. Is that clear? Is that clear? Is that clear? So prokaryotic cell, here you are talking about what? You are talking about the non-membrane bound organelle. That is your ribosome, the protein factory. But when we are talking about the eukaryotes, we'll have single membrane bound organelles, we have double membrane bound organelles, and even the non-membrane bound organelle is also there. And when it comes to the non-membrane bound organelle, students, you have the example of ribosome, right? You have the example of centriole in animal cell. That is again a non-membrane bound organelle. Okay, that is again what? A non-membrane bound organelle. So this part is clear? Done? Okay, great. So what are we going to start today? Today we will start with the plasma membrane. Today, what are we going to start? We are going to start with the plasma membrane, which is also known as cell membrane, which is also known as your plasma lemma. This is what we are going to discuss today. Already, beta, I have uploaded my handwritten notes here in this particular PDF that I will share in the Telegram group. So majorly my focus is to make that topic clear, that what exactly is present there, right? What is written in NCERT. I'll decode the NCERT lines as well for you. But then the notes part, I'll share in the PDF, I'll share in the Telegram group, or you will get that PDF link, and then, right, you can access that. Okay, got it? So let's focus on this part, plasma membrane or cell membrane. Even if you talk about the prokaryotic cell or the eukaryotic cell, we know that the plasma membrane will be there. Right? In both the cases, yes, we do have plasma membrane and the composition is quite similar, right? Like there are differences, but yeah, it's quite similar here. So now when I'm talking about the plasma membrane, of course, it is the, it is something which is helping the cell to interact with the surrounding. It is helping the cell to set the boundaries with the surrounding as well. It is helping the cell to pass the material, right, from it, right? So these are the things that we have to discuss regarding the plasma membrane. Right? These are the things that we have to discuss regarding the plasma membrane. So the very first thing that I would like to tell you here is that when it comes to the plasma membrane, it is selectively permeable in nature. What's that? It is selectively permeable in nature, or I would say differentially permeable, right? Selectively permeable or differentially permeable in nature. So what's the meaning of that, right? What's the meaning of that? So when I'm saying selectively permeable, so it will allow the entry or exit of substances across it. It will allow the entry or exit of substances across it as per requirement. Yes, so as per the need, as per the requirement, it will allow the entry or the exit of the substances. Okay, so it is selectively permeable. That is the proper word. Selectively permeable, that it as per the requirement, entry or exit of the things will be there. This is the one thing that you need to understand when you talk about the plasma membrane. Now, when we talk about the structure of plasma membrane, then what do we have? There are different models. There are different scientists, those who worked on the structure, then they decoded the things, then they explained the things. But the main things, main models that we have to discuss is, so firstly, I will start with Overton's suggestion. What am I going to start? Overton's suggestion. I will not start with the proper experimentation part here, that this is what Overton did, that is what Overton did. No, not. I'll give you the overview here, but I believe you people should know that. When it comes to the Overton suggestion, right? As per this, plasma membrane. For plasma membrane, I'll go for the short form, I'll write PM, okay? So as per this, plasma membrane is made up of, right? It is made up of lipids, right? The scientist Overton, as per him, plasma membrane is made up of lipids, okay? On what basis he said so? He observed that fat soluble things or lipid soluble things, right, they can cross the plasma membrane easily. That is what he observed. What he observed? He observed that the lipid soluble, fat soluble things can easily cross, right? They can easily cross the plasma membrane. That is what he studied. And on the basis of that, he suggested that plasma membrane is made up of lipids. Right now for you, because I know you guys know about it, that plasma membrane is made up of phospholipid bilayer. For right now for you, it's not a big deal. It's not a big achievement. But you know, back then, I would say the resources were not that available. We were not that well equipped, and somebody is observing details like that, right? It is a big deal. It is actually a big deal. I would say it is just like a breakthrough, because at least somebody was able to think that it is made up of something, and that something is lipids. That something is, right, that something is what? That something is fats. Is that clear? Is that clear? Is that clear? So as per this, plasma membrane is made up of lipids. Why? Because lipid soluble things can cross the plasma membrane easily. So that is what he observed. Done, students? Now, the second is Gotter and Grandel's suggestion. Do you know about it? No doubt the main model that we have to discuss is your fluid mosaic hypothesis. But yeah, I believe you people should know about it. So do you know about this Gotter and Grandel model or Gotter and Grandel suggestion? Yes, students? Do you have any idea about it? Gotter and Grandel model. Yes, Gotter and Grandel. So Gotter and Grandel model. So they were aligned with Overton. They were like, yeah, that's true. That's true that plasma membrane is made up of lipids, but they were the first one who mentioned that plasma membrane is a lipid bilayer, right? They said it's not a single layer, it's a bilayer. Now you must be thinking how? Of course, they studied the structure. They studied the structure of the cell, they calculated some surface areas, and on the basis of that, they provided their findings. Is that clear? Right? So they worked on RBCs and they found out that the lipid layer surface area is twice the surface area of the cells themselves. Are you getting my point? So Gotter and Grandel, as per him, plasma membrane is a lipid bilayer. So they worked on RBC's membrane. And you'll see, even in the NCERT, you'll read, right, that RBC membrane, right, the membrane of RBC, right, the membrane of RBC is studied in detail, right, for the explaining the structure of the plasma membrane. Can you tell me why? Yes? Yes? Anybody here in the class? Kindly answer my question. Yes? Why RBC membrane is used to study the plasma membrane? Anybody? Anybody? Why? Like, not just here in this model, in other models also, you people will see, right? You people are going to see, people are going to notice that they go for the RBC membrane. The reason is, when it comes to the RBC, you know that at maturity, it lacks nucleus. It doesn't have that major cell organelles as well. So it's very easy to, you know, extract that plasma membrane. Further, there is no organelle, no membranes are there, like no other major membrane bound organelles are there. So of course, it's easy to extract that plasma membrane. It's easy to study that plasma membrane, right? So it is providing a very simple structure for understanding that. That is why, right, they prefer the RBC's membrane. Okay, they prefer the RBC membrane. So Gotter and Grandel, that yeah, Overton is right. The membrane is made up of lipid, but the main thing is it's not the lipid single layer, but it is the bilayer. So they conducted an experiment, you know, they studied that particular cell, RBC cell, and when they checked the surface area, so it was even double than the surface area of the cell. So from there, it like this idea strike that this layer is basically, right, bilayer. This is a lipid bilayer. Then we talk about the next is Daniel and Davson model. Then we will talk about the Robertson model, right? So next is what? Next is Daniel-Davson model. Next is what, students? Yes, next is Daniel-Davson model. Okay, okay. Daniel-Davson model, right, beta? Yes. Okay. Daniel-Davson model, or you people can also call it as Tri-lamina model, or we can also call it as sandwich model, right? These are the names that we all can use for it. So as per this model, right, as per this model, see, tri-lamina, tri means 3, lamina means layer. So this is the hint that you people can pick, or the sandwich model. When we talk about the sandwich, this is what we have now, bread slices, and in between there is the stuffing, isn't it? Isn't it? That's what we have now, bread slices, and in between stuffing is there. Same is the case here. As per this particular model, right, as per this particular model, when it comes to the plasma membrane, yeah, even they were aligned that it is basically the lipid bilayer. Even they were aligned that it is basically what, students? It is basically the lipid bilayer. But as per them, there is a lipid phospholipid bilayer, but on its either sides, protein monolayer is present. But on their either side, protein monolayer is present. So it's like the phospholipid bilayer is sandwiched in between the monolayers of the proteins. Is that clear? Is that clear? Yes. Is that clear? So as per this particular model, right, as per this particular model, so as per them, there is the phospholipid bilayer, and here in the surrounding, what you people are going to see? This, right? So you will see that phospholipid bilayer is there, and this is the protein monolayer, and this is also again protein monolayer. So, and these proteins, they, as per them, they are the globular proteins. So that is what they suggested. That is what they suggested. Are you getting my point? So as per them, phospholipid bilayer, and both the sides are having protein monolayer. So we can say that phospholipid bilayer is sandwiched in between protein monolayer. Yes or no? They are sandwiched in between protein monolayer. Are you getting my point? They are sandwiched in between protein monolayer. Are you getting my point? Now, if I talk about the drawbacks there, okay, fine. Okay. So now look at the structure. One thing is clear. If something is lipid soluble, it can cross the plasma membrane, but not everything is lipid soluble. What about the things which are water soluble or lipid insoluble? How can they cross? Both the sides are having protein. How can they cross? So this model, right, failed to explain the transport. That's the main point. As I said, these models are not something, you know, very, 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 very important. But if you really want to understand, if you actually want to understand, yes, beta, if you actually want to understand the topic, if you actually want to understand it in detail, yes, beta, right? If you actually want to understand this, if you actually want to know this, right? So just keep it in your mind, right? Just keep the main points in your mind. So it failed to explain the transport. It failed to explain the transport of especially water soluble, or I would say lipid on insoluble substances across it, right? So it was not explained. Failed to explain active transport as well. Failed to explain active transport as well. So these were the drawbacks. Now the next is Robertson's model, which is also known as unit membrane hypothesis, which is also known as unit membrane hypothesis. Okay, so this is like here they didn't mention anything, you know, how can I say that? Yeah, so they didn't mention anything different or anything different from what we have studied in the Daniel-Davson model. The only thing was, like, see, let's say this. So in the Robertson model, again, as per this model also, right? As per this model also, one second. Here for this model also, RBC membrane studied. So they also studied the RBC membrane. So here, as per that, again, proteins are present on the either side, but as per this model, these proteins are fibrous proteins, right? Fibrous proteins. And overall, by this time, right, scientists, they were aware, as per them, the phospholipid bilayer is somewhere around 35 angstrom. Each protein monolayer is 20 angstrom or 20 angstrom. So that's the point, right? That's what they mentioned. That's what they discussed. Are you getting my point? That's what they mentioned. That's what they discussed. So you know about it? Yes or no? Done? Yes, please. Yes, please. Any doubts? Okay, done. So he studied what? RBC's membrane. So as per him, right, this is the thickness, like protein monolayers, they are having the same thickness, right? They are electron dense layers. This is the phospholipid bilayer. So the plasma membrane is in totality 75 angstrom. So as per this model. So actually Robertson, he observed it under the microscope, and what he found that there are there is a layer which is, you know, darkly stained, and there are layers which are lightly stained. That's what he noticed. Okay, he noticed the layer which are darkly stained and the lightly stained. Are you getting my point? Yes, darkly stained and the lightly stained. So like this is what he basically observed there, he found there, right? And as per this model also, like plasma membrane is tri-lamina. Plasma membrane is what? Plasma membrane is tri-lamina. Okay. Yes, done. So again it was like that there is one layer which is sandwiched in between two protein monolayer. So he also believed in the concept of tri-laminar model, just like Daniel and Davson. So his model, right, his model was just like a, you know, something that boosted the Daniel-Davson model as well. Are you getting my point? But again, if I talk about the drawbacks here, but yeah, the proteins, he suggested that they are fibrous proteins. So it is same like Daniel-Davson model. Right? They're just same like that. Here again, the transport part was not clear. So as per this model also, the phospholipid bilayer is sandwiched in between the two protein monolayers, right? So that's what we need to know. But now the model that we accept, the model which is given in our syllabus in detail is your fluid mosaic hypothesis. Which model is there? Fluid mosaic hypothesis. That's the model, or that's the hypothesis we have to study. Okay, that's the model or the hypothesis we have to study. So it is widely accepted. So it was given by Singer and Nicholson. Who gave it? Singer and Nicholson. Can you tell me the year here? Anybody here in the class? Anybody from my batch who can tell me the year here? That in which year? Yes? 19? Anybody? Yes? 1972. 1972. So it's important. It is a previous year question, right? So please write down. So basically, overall, even in the starting of the chapter, I told you, it is actually the electron microscopy, due to which we are able to study the structure of these membranes in that detail. Otherwise, it's not possible. So after the advent of microscopy, after that, that further invention in microscopy around 1950, we got to know about the structure of the plasma membrane. So Singer and Nicholson, they proposed a model, fluid mosaic hypothesis. And this particular model is most widely acceptable. Are you getting my point? So the year, the scientist name, both the things are important. What is important, students? Yes, Arsat, Sonu, what is important? What is important? Yes, HSP sir, long time no see. कहाँ गायब हो भाई? कहाँ गायब हो? क्यों गायब हो? HSP sir, long time no see. मतलब he didn't call. He's a very busy man. But when it comes to the YT session, then he's here. Always. Why, HSP sir? Where are you lost? See, I noticed you in first go. Right? So HSP sir, how are you? I hope you are doing great. I'll call you hopefully tomorrow. So guys, now come back to this topic. So Singer and Nicholson, 1972. It's a PYQ. It's a repeated question. So as per this, plasma membrane is quasi-fluid in nature. What is it? It is quasi-fluid in nature. What is the meaning of quasi? Quasi, somewhere you can understand like this, semi-fluid in nature. Like where, as per this model or as per this hypothesis, right? Thank you, HSP sir. Thank you so much. So as per this model, a plasma membrane is not liquid, it is not solid, it is, kind of fluid, and that fluid, in that fluid, your arrangement of your phospholipid and protein, it's not like that in the center you have the phospholipid, surrounding me you have the proteins. No, it's not like that. As per this model, that proteins are just like the icebergs in the sea of lipids, right? As per this model, I'll write the keywords here. All the notes are present here in the PDF only that I'll share in your Telegram group. Don't worry about that. So proteins are like the icebergs in the sea of lipids. Imagine there is a sea. You can imagine that sea as the sea of lipids. And at places you have the icebergs. So that icebergs are considered, like this is an analogy, right? So that icebergs are considered with the protein. They are basically considered equivalent to the proteins, and the sea is considered as the lipid. So that's the meaning here. So they are not talking about, a proper arrangement. So they are saying there is a kind of random distribution, right? The phospholipid bilayer is there, and at places in between you have the proteins, right? At places in between you have the proteins. So that is something, right? That is something which was discussed there in this fluid mosaic model. So you people can just have a look. Right now in detail we will study this. So you people can just see this. right? That is what they discussed here. So quasi-fluid, that is semi-fluid in nature. Proteins are like the icebergs in the sea of lipids. So these were the main points here. The arrangement was just like a mosaic when you look at it superficially, right? When you look at it like this now, you'll be like, okay, this is a cell. That is how the arrangement is. That is how the arrangement is. So now that's what we have to understand. Okay, so let's say this is a cell. Okay, you know that it is having the cytoplasm here. Yes or no? You know that it is having the cytoplasm here within the cell. Now in the surrounding, we have extracellular matrix. What do we have, students? We have extracellular matrix. You can call it as ECM as well, right? Or you can call it simply, this portion is your intracellular part. This portion is your extracellular part. These are the terminology that you all can use. So of course, if we have the cytoplasm here, we have the cytoplasm here. Cytoplasm is definitely going to have water without any doubt. Now in the surrounding also, we'll have water, right? Definitely we will have water. Now, if I zoom this portion, magnify this portion, if I look at it, so that's our plasma membrane. Let's say this is the intracellular portion. This is the extracellular portion. So now when you look at it, can I say that this side is facing the water and this side is also facing the water? This side is facing the water. This side is also facing the water. Can I say that? Can I say that? Now, on the basis of that, right, I'll continue my discussion. So, I will continue my discussion on the basis of that. So, one thing is clear that as per fluid mosaic model, right, it's not like that the proteins layers are arranged, you know, outside this phospholipid bilayer. No, proteins are present in between. They are having a kind of random arrangement here, right? It's just like proteins are the icebergs in the sea of lipids. So I hope this part or this thing is clear to everybody. Now I'll explain. Now we have to focus on the thing phospholipids here. We'll discuss its components, right? We'll break this topic into different components and then we'll combine everything, we'll discuss. So one thing that we know, that plasma membrane is made up of phospholipid. Plasma membrane is having the proteins. So firstly, let's talk about the phospholipids. Students, when we talk about the lipids, okay, lipids or your fats, they have two things. They have two things. Like they have, yes, they have fatty acid plus alcohol, and I'll be specific here. I'll talk about the glycerol because I am talking about the true fats here, right? Like lipids are of different types. You have the simple lipids, you have the compound lipids, you have the derived lipids. There are different types of lipids. So in simple word, right, just to make it clear, I'm simply telling you, right, guys, listen to me carefully. I'm simply telling you that we are talking about the lipids. Basically, we are talking about the fats here. What are we talking? We are talking about the fats here. See, my major focus is to clear this topic. So I want you people to listen to me very carefully. So I am saying that it is made up of phospholipids. So the word here is lipid. Lipid means fats, right? I'm talking about the true fats here. I'm saying fats are of different types. You have simple fats, you have conjugated or complex fats or complex lipids. You are having the derived lipids as well. So when you talk about the simple lipids, you talk about the true fats, and there you'll see that your fatty acid and glycerol will join. Who will join? Your fatty acid and glycerol will join. What is this glycerol? It is trihydroxypropane. Trihydroxypropane, right? It is even considered as the simplest fat. So trihydroxy, 1, 2, 3. It's propane, 3 carbons, trihydroxy. 3 times OH, OH, OH here. And here you people are going to see H, H. You just need to satisfy the valency here. So this is your glycerol. And it is going to form bond with fatty acids. So when I talk about the fatty acids, because it is a fatty acid, acidic group, you will have C double bond O, OH, and here you will have the alkyl group. So this COH and this hydroxyl here, there will be a bond formation in between. I'm not going to elaborate that in detail because we will discuss that in biomolecules. So ester bond will form here. Which bond will form here? Ester. So this is your trihydroxypropane having 1, 2 and 3 OH. If one fatty acid is attached, monoglyceride, 2 fatty acid di, 3 fatty acid tri. But what's the thing that I want to clarify here? That when your glycerol and fatty acid join, they are forming a true fat, right? This is about the lipids. This is about the simple lipids. Now, I am talking about the phospholipids here. So if I am talking about the phospholipids here, definitely it is going to have the phosphate group. That's what I am trying to highlight here. That definitely it is going to have the phosphate group. So the basic arrangement will be this only, that glycerol and fatty acid change, but along with that, phosphate group will also be there. And if I talk about the, right, most common phospholipid, then it is the example lecithin. Again, PY. Most common phospholipid is lecithin, having N group as well. Having N group as well. This is about the meaning of lipids. This is about the meaning of phospholipids. Now, when I look at the structure here, I'm talking about this round structure. I'm talking about something. I'm talking about this structure as well. So when you see this part, you call it as polar head. What do you call it, people? You call it as polar head. And this portion is nothing but the non-polar tail. This is the polar head. This is the non-polar tail. Is it clear? Polar head and non-polar tail. So when you look at this arrangement, I told you clearly, if I'm saying that this is the extracellular part, and if I'm saying that this is the intracellular part, so students, can we say that this head part is coming in contact with water here, and this head part is also coming in contact with the water here? Can I say so? Can I say so? That this portion here, this head region of phospholipid is facing the water. Here also this head region of phospholipid is facing the water. So if this is the case, right, if this is the case, then you need to understand this thing, that this polar head, it needs to be hydrophilic, and it is actually hydrophilic in nature. And what about tail, students? The tail here is hydrophobic. What's that? It is hydrophobic. So what is the meaning of these terms? Hydro means water. Philic means loving. So hydrophilic means it is water loving, and phobia means you have fear of something, right? So it is having the fear of water. So this is hydrophilic, this is hydrophobic. This is hydrophilic, this is hydrophobic. So when I look at this phospholipid here, when I look at this particular structure here, it is showing me both hydrophilic nature or the hydrophobic nature, polar part or the non-polar part. So that is why I call it as amphipathic, right? What's that? It is amphipathic. Yes, please tell me. What's that? It is amphipathic. Is that clear, everybody? It is amphipathic. It is amphipathic. So this is your phospholipid, right? As I said, it is amphipathic molecule because it is having both, right, this polar head which is hydrophilic, and I told you why it needs to be hydrophilic, and this is the tail which is hydrophobic, and I'm calling it as what? I am calling it as the non-polar tail. So when you look at this head part, here you will have glycerol, here you will have that so here you will have that glycerol, you will have in this portion the phosphate part here. When you look at this tail, right, when you look at this tail here, you'll see it is having the fatty acid chains. It is having the fatty acid chains. What does it contain? It is having that fatty acid chain. So any doubt here, guys? Any doubt here? So what are these fatty acid chain? I'll just give you one example. Like, see, it's an acidic thing. So C double bond OH is there. Rest you just need to satisfy the valency. You just need to satisfy the valency, right? So other than this C double bond OH, other than this particular carbon, we can have carbon 1 to 19 here in these fatty acids. Like you can be like this. If I have to give you the example, this is also a fatty acid. Now let's suppose you have added one more carbon here. This is also the fatty acid. This is also your fatty acid. This is also your fatty acid. So that's the example that I'm giving you. Okay. So this is the fatty acid chain. This is the fatty acid chain. Now when I look at this fatty acid chain, if there is carbon-carbon single bond, I am calling it as saturation. Right? Saturation. So at places we have carbon-carbon double bond as well. Okay, at places carbon-carbon double bond is also there in this fatty acid chain. So wherever there is a double bond, like see, simply it is going like this, or right, or you are making it like this. So wherever we will see the double bond, there will be a kind of kink or the bend in this fatty acid chain. There will be a kink or the bend, right? Right, there will be kink or the bend in this fatty acid chain. Let me show you the diagram here. Okay. Okay. So firstly, just look at this particular structure. This is the polar head, as I said, the phosphate group here. This is your glycerol here. This is the fatty acid chain. So saturated fatty acid like carbon-carbon-carbon-carbon single bond, and the place where unsaturation is there, double bond is there, we'll see a kink or the bending in the chain, right? We'll see a kink or the bending in the chain. Okay. Okay, that's how, see, it is arranged. So you can see that wherever unsaturation will be there, there will be the bend or the kink. So I'll show you this from my favorite, one of my favorite books, I would say. So see, like see, it is somewhere affecting the arrangement. I'll talk about it. We'll discuss it. See, wherever there will be the double bond, there will be a kind of double bond. There will be a kind of kink. There will be a kind of bond like this. There will be a kind of bond like this. Am I clear? Like bend like this, not the bond, bend like this. Understood? Understood? Yes, everybody, understood? Sure? Sure? So, right now just understand the arrangement. One by one, I'll tell you. And I'll explain you the part of each and every organ here. Each and every thing here. I'll explain you the function of each and every part here. Okay? Okay? Okay, done. So now in the plasma membrane, the arrangement is like this. Yes, the arrangement is like this. The arrangement is like this. Now you see the hydrophilic part is facing the water. Hydrophobic part is away from the water. So now in this plasma membrane, the arrangement is such a way, right? The hydrophobic tails, they are facing inwards. They are towards inner side, right? So they are forming a kind of water resistant system, right? Water resistant system they are making. So that, you know, water soluble things cannot cross through it. Cannot cross through it. I'll show you this point from the NCERT here, right? So you guys can see this thing here, that polar head towards the outer side, hydrophobic tails towards the inner side. This is going to ensure that non-polar tail of saturated hydrocarbon is protected from the aqueous environment. One thing is we are protecting that tail. And in addition to that, right, okay, so and it is also forming a kind of water resistant, right, water resistant or water resistant barrier as well. Is that clear? Water resistant barrier as well. Is that clear? Is that clear? Yes. Any other doubt? Yes, people. Any other doubt? Now, when you look at this particular structure, right, now there is another thing that we have to understand. So firstly, there is a water resistant barrier which will only allow the lipid soluble things to cross through it, right? Not the water soluble thing. This is one thing. Now, when you look at the phospholipid bilayer here, you people will see that for these individual layers, we use the word leaflet. Like this is your inner leaflet. This is your outer leaflet. Are you getting my point? This is your inner leaflet. This is your outer leaflet. That is also something that you need to understand. Okay, that is also something that you need to understand. Like if I'm talking about this particular diagram, chalo, I'll clear it and then I'll make it. let's suppose this, just showing you this, okay? Okay, so I am saying that this part is making your inner leaflet. This part is basically your outer leaflet. That's what I'm saying, inner and outer, inner and outer. Now, if you will read about the plasma membrane, you'll get to know that your plasma membrane is asymmetrical, right? Its structure is asymmetrical. Now, I'm trying to explain you that. See, when you look at this outer leaflet, inner leaflet, it is made up of different phospholipids. And this one is having, this one is also having different phospholipids. Like they both are having different phospholipids. Are you getting my point? The phospholipids here, the composition of phospholipid, right? The composition of phospholipid is not same here. Is that clear, everybody? Is that clear? Like if I have to give you the example, so here I will say, I'm going to say that here you have phosphatidylcholine. These are the name of the phospholipids. You have your sphingomyelin, but yeah, there is no need to remember them. These are different types of phospholipid. This is just for your knowledge. Okay, sphingomyelins, phosphatidylcholines, right? And here you guys can have, you know, phosphatidyl inositols, phosphatidyl ethanolamide. So that's what we have. This is about the composition here, right? This is about the composition here. Now, there is something which you really need to understand, that is the fluidity here. So before fluidity, yeah, one thing is this about the asymmetry. Secondly, outer side of the leaflet, outer leaflet, which is facing the extracellular matrix, here with the lipids, you people will see, right, lipids, proteins are attached, like oligosaccharides are attached, glycolipids we have, glycoproteins we have. We'll talk about it. So that's why there is the asymmetry. Now, we need to discuss something known as fluidity. In the starting itself, I told you your plasma membrane is not a solid, right? It is quasi-fluid in nature, it is semi-fluid in nature, and that fluid nature is very important, because that fluid nature of plasma membrane is important for the growth, is important for the maintenance of the shape there. It is important for that fluid nature, is important for the interaction. That fluid nature is important for the movement of substance across it. So yeah, plasma membrane, it needs to be, right? The plasma membrane, it needs to be fluid-like. Now, what is this fluidity? Actually, in the plasma membrane, these phospholipids, they can exchange their position with their neighboring phospholipid, right? I'm talking about the leaflet. I'm not saying that phospholipid is going from one leaflet to another. No. Right now, I'm talking about the fluidity, right? Fluidity. So I am saying that these phospholipids, right? These phospholipids can exchange position with their neighboring phospholipid, right? They can just exchange their position. In the same leaflet here. Plus, not just phospholipid, your proteins as well. Proteins also, like the proteins which are present. There are some now which are, you know, fixed. I'm not talking about them, others. So they can exchange the position, right? They can exchange the position. Are you getting my point? So this phospholipid can move to this side. This phospholipid can move to this side. Even the proteins can also exchange the position. Are you getting my point? Even the proteins can also exchange the position. The phospholipids and the proteins both can exchange the position with their neighboring phospholipid in the same leaflet. So this property is fluidity. This property is fluidity, which is an important MCQ, important PYQ, right? This property is giving this plasma membrane this fluid-like nature, right? That is something you need to understand here. Are you getting my point? That is something you really need to understand here. Is that clear, everybody? Is that clear, everybody? This is the fluidity, right? So helping the cell to grow, helping the movement of the substance. If your plasma membrane will be solid, how the exchange will occur, how the growth will occur. So yeah, that is important. Now, there is one more thing that is known as flip-flop. There is one more movement that is known as flip-flop. It's a very rare movement. Do you know that? It's a very, very, very rare movement. Why rare? Because it is not taking place usually. And I'll give you one example, like once I was studying it regarding the cancer cell. When a cell becomes cancerous, they mark it. Okay, that's why the cells, defensive cells of our body, right? That's why the immune cells of our body, they are able to detect if some cell is not working well or not. You know, in our body, programmed cell deaths, they all are present in our body. So basically the point here is there is a very rare movement which we are calling as flip-flop. In that flip-flop movement, what's going to happen? See, right now in fluidity, the phospholipids are exchanging position with their neighbors, right? Or proteins or phospholipids are exchanging position with their neighbors. But when there is a jump of phospholipids from outer leaflet to inner, right? A phospholipid which is basically the part of inner leaflet, inner to outer, right? The part of inner leaflet. Let's suppose there is a phospholipid which is the part of what? Which is the part of inner leaflet, jumped to the outer, right? So this is a very rare movement. It takes a lot of energy, and yeah, in some cases it also indicates the cancer. It's also a marker of cancer, that this cell has become cancerous, not working well. That's why changes like this is occurring, and that is going to give a signal, that is going to induce a signal that there is a requirement of the cell death of that particular cell. Are you hearing my point? So that's something that you people need to understand. Okay, that's something you people need to understand here. Okay, so this is the flip-flop rare movement, right? Phospholipid of inner leaflet jumps to outer. Understood? Done? Yes. So fluidity allow a cell to, stretch under stress and to reseal if disrupted. So it allows a cell to stretch under stress and to reseal if disrupted. Got it? If disrupted. Got it? So that's how it works. And I told you flip-flop is a rare movement. Okay? It's a rare movement. Is that clear? Now, the next thing that you have to understand is, then I'll come to the saturation, unsaturation part. Next thing is about the proteins. Okay, students, next thing is about the proteins. So when it comes to the proteins, so on the ease of extraction, on what? On the ease of extraction, you are going to talk about the peripheral proteins, which we can easily extract, also known as extrinsic proteins, and the another category is of integral proteins, also known as intrinsic protein. Yes, everybody. So this one is peripheral or extrinsic, integral or intrinsic. So it is on the basis of ease of extraction. So it is on the basis of ease of extraction, the protein which you can extract easily, right? The protein which you can extract easily. So peripheral, or here you have integral. So when you are talking about the peripheral protein, they are on the periphery. You can, without disrupting the cell membrane, you can remove them. So an integral protein, if you need to take them out, obviously you have to disrupt the cell membrane. So that's what you have to understand. So before going forward, let me tell you, overall in a cell, the ratio of this lipid and protein, it varies in the cell membrane. It varies. It literally varies when it comes to the ratio of the lipid and protein. Like in the case of RBC, in the case of RBC's membrane, it's 52, near about 52% are proteins 40% are near about lipids. So it varies. It varies. And when you talk about these proteins, they are going to provide, right? They are going to provide specificity to the cell membrane. Do you know that? They are going to provide specificity to the cell membrane, right? How? I'll explain you. Firstly, I'll tell you about the arrangement. So we'll take the help of this diagram, and with the help of that, I'll tell you. So you look at this diagram. There are some proteins which are basically crossing this plasma membrane, right? There are some proteins which are basically crossing this plasma membrane. Can you see that? Can you see that? These are obviously the example of intrinsic protein. And what sort of intrinsic proteins are they? They are the tunnel proteins. They are the channel proteins, because they are the transmembrane proteins. They are traversing this plasma membrane. They are crossing this plasma membrane. Right? They are traversing this plasma membrane. They are crossing this plasma membrane. So this is basically the tunnel protein. These are basically the tunnel proteins. Are you getting my point? Just like tunnel. Some will be located on the surface superficially like this. They'll be the extrinsic protein. So these proteins are definitely going to provide the specificity, right? The specificity to the cell membrane. And students, around 70%, right? Around what? Around 70% of the proteins, right? Around 70% of proteins are integrals. Do you know that? They are integral proteins. Are you getting my point? Around 70% are what? They are integral protein. They are embedded here in the phospholipid matrix, and they are also, like, if I have to give you the example, I'll give you the example of glycophorins as well. Okay? Okay. So now, because when you talk about these proteins, they are having the scattered nature, right? That's why, right? They are having the scattered nature. Proteins are scattered here in this phospholipid layer, right? Proteins are basically scattered. So this is actually giving it mosaic-like appearance. It is actually giving it mosaic-like appearance. Okay. So now if I talk about the proteins, what is the role of proteins here? Different roles are there. Like if I talk about these channel proteins, as the name is suggesting, channel proteins, right? They'll allow the entry or the exit of the things which are lipid insoluble. Let's suppose if there is any charged thing, right? If there is any charged thing, if there is something which is lipid insoluble, of course, it depends upon size as well. It depends upon the charge on that particular thing as well. So they are able to cross it. I'll give you one very common example. You might know about the porins, pore-like channels on plasma membrane, pore-like structures on plasma membrane, porins are there. So porins, if I call them as aquaporins. Aqua means water. So aquaporins, you know, water movement will be across it. Okay, so even ammonia, it also crosses this. Are you getting my point? So that's how the things can cross. So they are acting like a channel through which things can cross. They are helping in that transport. Okay, they are helping in that transport. So overall, if I talk about the proteins, you people will see that proteins, you know, they are also acting like receptors. And with them, the suitable thing, right? Their suitable complementary part will join. They'll, you know, help in initiating some processes. We discuss that when we discuss the mechanism of hormone action, right? We talk about the fertilization, students. We discuss that sperm and ova, they have the compatibility, they'll fuse. This is going to happen. That is, of course, there are proteins on their surfaces, right? That will, you know, make it species specific proteins are there, right? When that sperm and ovum belongs to same species, specific proteins, you know, proteins, receptors, they all are present. They are having that. So that's why, on the basis of that, there will be the compatibility. I'll give you one example. We know the endocrine system in our body, endocrine glands in our body. We know that they will release their hormones in the blood. So how that thyroid stimulating hormone is clear, that I have to act on thyroid only. Because for that, the suitable receptor is on that particular gland, right? So that receptors are what? They are also proteinaceous. So basically, that is why I'm saying that these proteins are providing specificity, right? These proteins are providing specificity to the plasma membrane, because all the different types of cells are having their different proteins as per their function, as per their needs. Okay, as per their function, as per their need. So that is something you have to keep in your mind. Okay, that is something you have to keep in your mind. So these proteins here, right, they are acting like enzymes, they are acting like channel proteins, carrier proteins. Some will allow the entry and exit of substance, you know, passively. Some require the energy here. So that's something you have to understand. Okay, that is something you have to understand. Okay. Okay, now from this diagram itself, I'll explain you that plasma membrane also contain oligosaccharides, means carbohydrates, right, particularly here on this outer leaflet. So if they are attached to the lipid part, they'll make glycolipids. If they are attached to the protein part, they are making glycoproteins. So these glycolipids, these glycoproteins, they are very important. They help in cell-to-cell recognition, right? They even help in, you know, differentiating a self-cell, like as a cell of our body, right? Differentiating the own cell of our body from a foreign cell, if there is any in our body. So that is how they work. That is how these work, right? Now, the next part is cholesterol. Cholesterol. So, cholesterol, the example of derived lipid, it is present in animal cell membrane. It is present in animal cell membrane. Where is it present, students? It is present in animal cell membrane. What? Cholesterol. Cholesterol. Okay. This cholesterol, it is absent in plants. Plants are not having cholesterol, but they are having some other sterol. Are you getting my point? Some other sterol. Basically, they are the steroids only, na? If they are alcoholic steroid, this is sterol. If they are ketonic steroid, they are sterones. So it is not there in the, it is absent in plants, but it is present in animals. And if you remember, in bacterial cell membrane, in bacterial cell membrane, I clearly told you this thing. They lack cholesterol. They lack cholesterol, but are having some other pentacyclic compound for stability. Pentacyclic compound for stability, for providing stability to the membrane. And if I have to give you one example, I'll go for hopenoids. I'll go for hopenoids. What will I do? I'll go for hopenoids. Understood? Yes. Understood? Sure? So, cholesterol is very important. Cholesterol in the plasma membrane is very important for the stability part as well. Okay? Because this is, see, it is inserted between phospholipids and it is more stiff than phospholipid. So, of course, if it is more stiff than phospholipid, so provide stability to that structure, right? So do what? So provide stability to the structure. Are you getting my point? So do what? So to provide stability to the structure. So do what? Provide stability to the structure. Are you getting my point? Are you getting my point? Yes, everybody? Yes, everybody? So how this cholesterol will help? I'll show you. See this thing. Not this one. This one. Right? This one. You have to see. Look at this cholesterol part first. Cholesterol within the animal cell membrane. What that cholesterol will do? This cholesterol, it reduces the membrane fluidity at moderate temperature. When the temperature is moderate, not high, not low, it will reduce the membrane fluidity by reducing phospholipid movement. So it is maintaining that. Like let's suppose if the temperature is very down, the things can get solidified. It will prevent that plasma membrane to get solidified. If the temperature is very high, it will more like a liquid. It will stop that as well. That's how the cholesterol provides the stability. Because it is more stiff than phospholipid, it is capable of doing that. Okay, it is capable of doing that. Are you getting my point? So by reducing the phospholipid movement, but at low temperature, it hinders, like it stops solidification by disrupting that, compact packaging. That is how the cholesterol help. And now, one more thing regarding the unsaturated or saturated hydrocarbons. You just look at it. If in the plasma membrane, unsaturation is not there, or if it is less, if unsaturation is not there, right, beta? Yes? Yes? See this part here? So if unsaturation is not there, saturation is there. Can you see that? Just look at the diagram. It is compact. It is viscous. It is more viscous. Right? Viscous. So if somebody is going to ask you, what happens to the plasma membrane if its saturation increases? It will become viscous. It is going to be viscous. It is going to be more solid, like if you increase it more. Right? But what happens when there is the unsaturation? So basically there should be a balance. So unsaturation will do what? The bends, the kinks will be there. So you can see the gaps. It's not very compactly packed. So it will be fluid-like. So unsaturation prevent packing, enhancing membrane fluidity. Saturated hydrocarbon tails, they pack together. They increase membrane viscosity. They increase membrane viscosity. Is that clear? They increase membrane viscosity. Is that clear? Is that clear, everybody? Sure, sure. So there are some plants which are having this adaptation that as per the seasonal changes, they can increase the level of unsaturated or saturated hydrocarbons in their plasma membrane. Okay? Okay? Done? Sure? Done? Right? So this is about the plasma membrane. Now we will talk about the transport across it. So when it comes to the transport, the very first thing is passive transport, and the second important thing here is the active transport. So transport across membrane is very important, and yeah, this model is able to explain that. This model is able to explain that. Passive, in the case of passive transport, the movement of substance, right? I'll write the keyword. The movement of substance is from its higher concentration to its lower concentration. Are you getting my point? From its higher concentration to its lower concentration, that is down or along the concentration gradient. That is down or along the concentration gradient. Gradient, you understand? Difference, right? Like at one place something is more, at one place it is less. So it will be high to low, right? And that will be down or along the concentration gradient. Along the concentration gradient, down to along the concentration gradient, but without using ATP. So here there is no usage of ATP, that the energy currency, things are moving passively, right? Things are moving passively. It's like you are basically coming down from a hill. So the movement will be very smooth, high to low, right? The energy will not be used like that. Now, active transport, substance move against the concentration gradient. What's the word here, students? It is against the concentration gradient. What's the word here, people? It is against the concentration gradient, that is from low to high. So if you have to do something against the concentration gradient, like if let's suppose there is a cult, right? People are following something, and now you want to follow something else. Yes, bache? Right? So substance move against the concentration gradient, that is from low to high. So if all are following something and you have to do something else, you have to put efforts for that, you have to fight for it, something like that. So low to high. So obviously ATP is used here. So I remember it like this: A for active, A for ATP, A for against, right? And that is through carrier proteins, right? Through that channel proteins or carrier proteins, because they will not be able to cross the plasma membrane directly. So channel proteins are required. So this is the active transport. So guys, when I talk about the passive transport, right? When I talk about what? When I talk about the passive transport, right? You all know many examples. I hope you know about the diffusion, right? Diffusion. Things will move from their higher concentration to lower concentration. In 9th class also we used to read it, without expenditure of energy. Then next is facilitated diffusion. Facilitated, right? The substance will be facilitated, like carrier proteins will be provided. It will not cross the plasma membrane directly. It will not cross the plasma membrane directly, but rather through the channel proteins. Through these channel proteins it will cross. Through these channels it will cross. So facilitated diffusion it is. So ultimately it is the diffusion. The things will move passively, right? The things will move down the concentration gradient. But here we are not talking about, it is a simple diffusion. Things are crossing the plasma membrane directly from higher concentration to the lower concentration. But when there is the facilitated diffusion, you need the channel protein there. You need the tunnel protein there. Is that clear? Is that clear? Now when you talk about the osmosis, osmosis is also a special case of diffusion only. So if it is a special case of diffusion, students, do let me know. If it is a special case of diffusion, so obviously the movement will be down the gradient. But here, water, or I would say solvent, move across semi or selectively permeable or differentially permeable membrane, right? It is the solvent. You have to be specific here. You cannot just say solute. It is the solvent. It is the solvent. Water, or I would say solvent, right? Water, or I would say solvent here, right, students? Right, students, that will move across, that will move across semi-permeable or selectively permeable membrane from its higher concentration to its lower concentration. Like the movement of water from will be there from water's higher concentration to water's lower concentration through a semi-permeable membrane. Like for an example, this is a solution. Solution A, solution B. I'm saying that solution A is having 10 gram solute, solution B is having 5 gram solute. Okay, now you tell me about the water movement. Will it be from A to B or it will be from B to A? What should be the answer here? Yeah, tell me quickly. Yes, please. Wow, A to B. Are you kidding me, guys? Are you kidding me? The water movement will be from B to A. I just said there in the osmosis, there is a movement of water, or I would say solvent is crossing. We are not talking about the solute. So here you have less solute means more solvent. More solvent, and in between these two you have this semi-permeable or selectively permeable membrane. 10 gram solute means in comparison to B, it is having less solvent. So movement of water needs to be there. Movement of water needs to be there. So it will be high to low. It will be high to low. So the water movement will be from B to A, right? So the water movement will be from B to A. Is that clear? So kindly pay attention. Right? Kindly pay attention. It's important. It's very, very, very important. So please pay attention, okay? So let's revise it from the NCERT once. So we are saying that the detailed structure of the membrane was studied only after the advent of electron microscopy. When? In 1950s. Just now I told you. So meanwhile, chemical studies on cell membrane, especially RBC. Now you know why RBC. So this is actually the decoding of NCERT, okay? So it enabled the scientists to deduce the possible structure of the plasma membrane. So these studies showed that the cell membrane, is mainly composed of lipids and proteins. Cell membrane is mainly composed of lipids and proteins. So the major lipids are phospholipids arranged in a bilayer, right? They are making 2 layers. So lipids are arranged with the polar head outwards, already discussed that, right? Non-polar tail towards inward. So that hydrophobic tail will be protected as well. And in the plasma membrane, cholesterol is there. So this is not the complete information. You know it very well that this cholesterol is present in animal cell membrane. It is present in animal cell membrane. Am I clear? So later biochemical investigations, they revealed that cell membrane also having proteins, carbohydrate, the ratio varies. It depends upon the cell type, students. So erythrocytes are having approximately 52% proteins, as I said, and 40% lipid, students. Okay. So next is on the ease of extraction. Membrane proteins can be classified as integral or peripheral. So peripheral will lie on the surface of membrane and integral will be totally or partially buried. Okay, right? So the next part, and this is the diagram from the NCERT which you really need to remember. Okay, so the next part is that improved model for this structure is given by Singer and Nicholson, 1972, a repeated PYQ. This is again a repeated PYQ, right? So they gave the fluid mosaic model, which is widely accepted. Okay, so as per this, your plasma membrane is quasi-fluid in nature, that is semi-fluid in nature, my dear people. And because of that, lipids can move laterally, right? So this lateral movement is actually known as fluidity. Fluidity. What is it? Fluidity. Oh, don't tell me I have mentioned it as fluidity. Okay, so this is fluidity. Okay, so fluid nature of the membrane is very important because it will help in the growth of the cell, formation of intercellular junctions, like the one which will connect 2 cells together, then secretion, then endocytosis, then endo. Students, endo means inside. Like in class 9th, we used to read it, amoeba. Amoeba used to engulf the food. How? By endocytosis, the invagination of the plasma membrane. Plasma membrane will invaginate. Plasma membrane will form a cup-shaped structure around the food vacuole, around the food, and in the form of vacuole, that food will surrounded by the vacuole, then the food will enter in the cell. Endocytosis. So yes, endocytosis, right? So basically, this is something like we are talking about something which is entering in the cell, endocytosis. If we are saying exocytosis, something is going out from the cell, right? Out from the cell, exocytosis. This is your cell vomiting, right? Cell kind of eating. And here, if I have to be specific, so phagocytosis, when there is the, solid substance will get enter in the cell. Then there is the penocytosis as well. So penocytosis, my dear students, is cell drinking, right? It is the cell drinking. So pinosomes will form, in which there will be that liquid particle that will enter in the cell. So one is the in which solid particle will enter, in which liquid particle will enter. So pinocytosis, right? Cell drinking it is. Next, fluid nature is one of the most important function that plasma membrane helps in that transport. So it is selectively permeable, my dear students, right? So we know that if something is moving without the usage of energy, this will be passive transport, right? And when neutral solutes, when you people discuss about the neutral solutes, they'll cross the membrane by simple diffusion. Neutral, it is no charge, simply it will go along the concentration gradient. It doesn't need any channels as well, right? So water can also cross. This is osmosis, we have discussed that. But if there is something which is charged, right, polar molecules, they cannot cross the membrane directly, so they need the channels, they need the facilitate, they need to be facilitated, right? So few ions or molecules are transport against the gradient. So it is the active transport. So here in the NCERT, they have given us the example of sodium potassium ATPase pump. Where are we going to study it? We'll study it in the nerve cell, right? Where we will see that against the concentration gradient, sodium and potassium ions will be exchanged, okay? Will be exchanged. So this is all about the plasma membrane, right? You revise it properly. Okay, then we have to study the cell wall. So my next class will be there on Monday. Okay, then again I'll keep it its timing. I'll keep it at 7 or 8, right? So we'll stretch it and we'll finish the rest of the part. Okay, students. So I hope you liked the session. I hope you enjoyed it. Now you know what you have to do. If you want to be the part of the batch, and you should definitely be the part of the batch because they are going amazingly, right? The test, the practice, everything is there. And you know that due to that we have our Independence Day, and for that also, you people are having amazing deals, amazing offers. So what you are supposed to do? Yes, what you need to do, guys, tell me. You just need to go to the description box of this particular video, right? You just need to go to the description box of this particular video, right? And see, you have to click on this particular link. The coupon code will be already there and you'll get the lightning deal. And here, this is the link where you can just get the PDF of, right? And here you will get all the PDFs actually. So you have to select your category. You have to click there. Okay. And then see, this is how they're going to ask. You just fill up your details. Let's suppose I filled this and I'll move like that and I have to fill my detail and you will get all the PDFs. Okay. Okay. So this is all about today's session. And as I said, you people will get the PDF. Okay. So keep studying, keep revising. Done. Keep studying, keep revising. So see you all soon. So till then, take care. If you like the session, do let me know in the comment section.